Okay, so in that first session we went through a breeder's guide to genomics, so a guide as to, I guess, what we must do, should do or could do with regards to genomics. This next session we just want to spend a little bit of time talking about, well, how do we extract value from genomics? So when we take a DNA sample and we send that off to the laboratory, the results that we get back, say for a product like Angus GS, if you like, which is a genomic product which has around 48,000 SNPs or single nucleotide polymorphisms. So that DNA test goes in and reads the animal DNA and returns a result at each of those SNPs for basically a series of A's and B's. So at each particular SNP, the potential results that we can get for each animal are either an AA, an AB or a BB. And so we get a result back from the laboratory of basically a series of 48,000 A's and B's. And how do we extract information from that in terms of predicting the animal's breeding value? And I want to spend, this is really where the genomic reference population comes in, so I just want to spend a little bit of time really stressing to you that the value we obtain from genomics relies on having a really well-recorded, relevant genomic reference population, and particularly for hard-to-measure traits. So what do I mean by genomic reference population? We're talking in this sense around a population of animals with both genotypes or DNA information and phenotypes or performance information. And the value that we extract from that DNA result that we get back from the laboratory really depends on the existence of this really relevant and well-recorded genomic reference population. So to explain that in a little bit more detail, I just want to go through and reflect, I guess, first of all, some of the fundamentals of genetic evaluation. So if we think about how we've been running a genetic evaluation for the last 30 years or so, the basic method by which that genetic evaluation worked is by we had performance information on the animal in which we're trying to predict its genetic merit. We've got performance information on its relatives, and we use both those sources of information to go through and predict what that animal's breeding value is. So if we look at this diagram in a little bit more detail, and we look at how we used to determine, I guess, the animals to which the animal was related to, and therefore use that performance information to estimate breeding merit. We have the animal itself, we've got animals in its pedigree and the performance information on those, and also their descendants and the performance information on those. And we potentially had progeny of the animal as well and performance information on that. So we took all those different sources of information, the information on the animal, the information on its pedigree, the information on its progeny, put that together and estimated breeding value. And the way we determined those relationships between animals was really, the genetic relationships between animals was to do with the pedigree information. Now when we're in a genomics world, we've got exactly that same situation. So we've still got the animal itself, we've still got its pedigree and the performance information on those animals. We've still got its progeny and the performance information on those, but now we have an additional source of information which we can use to estimate the animal's breeding value. And this is the genomic reference population. So now, through DNA, we've still got the relationships based on the pedigree, but by combining in the DNA information, we'll know that this animal, even though it might not have any direct pedigree links to the animals in the genomic reference population, it will share some genes in common. So if the animal has DNA, and we've got the animals out here in the genomic reference population with DNA, we can use that to determine the relationship, the genetic relationship between that animal and that genomic reference population, and start to use the performance information which has been collected on the genomic reference population to help contribute and estimate the animal's breeding value. So we suddenly now get an additional source of information with which we can use. And so when we're looking at genomics, the reason we can now better estimate breeding value is twofold. The first is we can now start to use performance information on animals which were previously unrelated by pedigree, but now with the benefit of having DNA information available, we can understand that genetic relationship between those animals and the animal with which we're trying to estimate breeding value and use that performance information as an additional source. We also now, if the animal has DNA and the animals in its pedigree or its progeny have DNA, we can more precisely understand what the true relationship is between those animals and weight the information on those animals more precisely to better predict the breeding value. So due to those two reasons, I guess we can use genomics to better estimate the breeding value of animals or do it with more accuracy. Now some of the key things that, if you like, there's the standard saying and slogan which people understand of oils ain't oils. Well, genomics ain't genomics. So the value that we get from that genomic reference population really depends on a number of factors. So the first, of course, is the size of the reference population. So in general, the larger the reference population, so the more number of animals with both phenotypes and genotypes, the more value we can extract from that genomic reference population to estimate an animal's breeding value. And we'll talk about that a bit more. It's also the closeness to which the animal that we're trying to predict the breeding value of, how closely related it is to the animals in the genomic reference population. So if we've got an animal that is more closely related to the animals in the reference population, then that provides a lot more information. It's a lot more informative as to what the animal's breeding value is, compared to if that genomic reference population is on quite distantly related animals. So particularly if we look at genomics in Australia, whether we have an Australian reference population with Australian animals, or if we've got, say, a North American reference population or a European reference population, or one from Great Britain, those animals will be more distantly related, and that genomic reference population will be far less value and provide far less information in estimating the animal's genetic merit in Australia. Likewise, the value of the phenotypes that have been collected on those animals in the reference population for estimating genetic merit. So in that way, if we've got traits which are lower heritability, so a less proportion of the differences that we see between animals can be attributed to genetics, we need a lot bigger reference population to get the same level of accuracy or prediction of genetic merit. And another one is just the relevance of the production systems in which the phenotypes have been collected. So in that case, we want that performance information to be collected in production systems which are similar to the production systems in which the progeny of the animal with which we're trying to predict the breeding value are going to be used. And again, when we start looking at evaluating the genomics genomics ain't genomics, then if we're looking at that reference population, we really want it to be performance information collected in Australian production systems that are similar to the ones that we're trying to breed the progeny for, compared to, say, production systems in overseas countries. So if we break that down and evaluate and just interrogate it a little bit further, here we have a graph which just demonstrates the influence of the size of the reference population on how the accuracy of genetic prediction. So across this bottom axis here, we have the size of the reference population. So you can see the increasing size from say 2,000 animals across here to 20,000 animals, versus the accuracy with which we can predict an animal's genetic merit. And you'll see a couple of key points. The first is, as we increase the size of the genetic reference population, then we can predict breeding merit with more accuracy. But that's not linear. So we go through and we get an initial really big increase up here to probably around 10,000 animals, and then that's the value of having additional animals in the reference population starts its law of diminishing returns, it starts to flatten out. The other thing that you'll note is there's a number of different lines on that graph, each depending on the heritability of the trait. So as we said before, if we've got a trait which is lower heritability, we need a larger reference population for the same accuracy of prediction compared to a higher heritability trait. Now, a lot of our production traits with which we're trying to use in our breeding programs have a heritability of around 0.3. So if we look at this green line in the middle, what we're trying to do in the case of most of our building of our genomic reference population is really targeting a reference population with about 5,000 to 10,000 animals. So we're starting to get up here to an accuracy of prediction of around 50%. And then we need ongoing maintenance of that reference population to ensure that it continues to deliver the same level of value. So that's really what we're trying to do. Now in the Angus breed, what's the size of the genomic reference population? So when you, as members of Angus Australia, send DNA samples to us, we send it off to the laboratory, we get a result back, and we include in the Trans-Tasman Angus cattle evaluation, what's the size of the genomic reference population that we use? And this slide here just gives you a graph based on, say, the mid-July 2019 Trans-Tasman analysis for all animals born after 2011. It shows you what the size of that reference population is. So if we look in terms of the total animals, the total size of the reference population now is around 60,000 animals. And the black bar on this graph shows the animals which have been genotyped in industry or member herds. Versus the red is the animals which have been genotyped within the Angus sire benchmarking program. So first of all, that first bar on the graph, the total animals, are just the animals which have been genotyped. Across the different bars, you can see, well, of those animals, what performance information has been collected on them. So what's the size of the genomic reference population for those traits? And you'll see certainly for some of the more routinely measured traits, so things like 200-day weight, 400-day weight, birth weight, etc., even 600-day weight, we've now got a reference population that's well over 20,000 animals. So it's a really useful, relevant, well-recorded reference population for which we can extract a lot of value. Now, of course, they're not the traits with which we really need genomics for, because they're the traits which we've been collecting performance information on and selecting on for some time. The real value of genomics becomes from the hard-to-measure traits. So we start looking at the abattoir carcass traits, the female fertility traits, mature cow weight, etc. And if I break those traits down a little bit more, here you can see the size of the genomic reference population that we're currently using. So the couple of things you'll notice on that graph, one is that the size of the reference population is a lot smaller. So we're starting to look now at around 3,000 animals in that genomic reference population. Now say that's a far greater size than what a lot of other beef populations have available to them. So it's a really useful size, but it still needs to be built. And that's why the funding and the continued support of the sire benchmarking program is critical for us as an Angus breed going forward to get the most out of genomics. The other thing that you'll note is around the bars are all red now. So all the data with basically the performance information which is building this genomic reference population for use by Angus Australia members is basically being collected through the Angus sire benchmarking program. So that's where that program is really delivering a lot of value. Now those traits on those graphs are really the traits for which we currently have breeding values produced. One of the other things that we're starting to see is building a genomic reference population for other traits which we probably haven't even started to think about selecting on or aren't routinely selecting on in our breeding programs. So we'll see here just some additional traits which have been collected in the Angus sire benchmarking program for things like immune competence, and our next session we'll go through and talk about some of the early work to produce research breeding values for that trait, through to some better indications of eating quality, so MSA index, ossification, etc., some fatty acid profiles, methane emission, down into some of the more adaptability traits such as coat score, etc. So in time, that's really where we're going to get the major benefit at this stage as we start to build the genomic reference population for those traits. And you can see now across some of those traits, we are starting to get to a level where we're hitting that target of that 5,000 to 10,000 records, where we'll start to get really useful accuracy of prediction. So the message I really just want to stress to you is that, first of all, as we said, that the value we obtain from genomics is determined by having a really well-recorded, relevant genomic reference population. And in that sense, if you're looking at different genomic products from different people, it's really important to be considering what that genomic reference population is. And secondly, as Angus breeders in Australia, you're quite well positioned in that through the foresight of the Angus Australia board and management many years ago in starting the Angus sire benchmarking program, the sire benchmarking program is now delivering a really relevant, well-recorded genomic reference population with which we can build on and really get maximum value out of genomics. So they're couple of key messages. Now, of course, that goes through and raises a couple of questions, and some of the things we've been talking about at Angus Australia recently at a board and senior management level is around, well, how do we fund the Angus sire benchmarking program, or the development of the genomic reference population on an ongoing basis and do that in a sustainable manner? And secondly, how do we get equitable access to that genomic reference population? So by people who've been involved with Angus Australia for a long time and contributing a lot to that genomic reference population versus others who may just recently join, start to test their animals, how do we ensure that they get equitable access that reflects the different investment that they've had in building that genomic reference population? And there's just a couple of different things that I want to share with you around this. One thing which has been in place for some time is that when you order DNA services from Angus Australia, a component of the fee that you pay is for the ongoing maintenance and building of that genomic reference population. So when at the moment you pay your $51.45 for either an Angus GS or an HD50K or the Angus Ultra Low Density at $40, around $5 of that fee goes and contributes to the building of that reference population. Now that doesn't go anywhere near to covering the cost of that reference population. So at the moment that covers around 10% of the total cost to Angus Australia. The rest of that cost is currently covered by funding from Meat and Livestock Australia through the MLA Donor Company scheme and also from other Angus Australia income sources. But in time, as genotyping grows, then I guess the model is in place to really try and fund that on a sustainable way and contribute far more than that 10%. In terms of equitable access, then we have at an Angus Australia level really been mindful, well, then there are some herds out there who are investing a lot in collecting these hard to measure traits on genotyped animals, and therefore it's not really equitable for them to be investing on farm and doing the hard yards, but then also being required to pay that $5 kind of contribution to the maintenance of the reference population. So as of the start of 2020, so as of January 2020, there will be a rebate scheme introduced so that any member who collects information on hard to measure traits on genotyped animals and that performance information that they collect meets certain guidelines so that it's useful for genetic evaluation and for the genomic reference population, will be eligible to receive rebates by way of a credit to their Angus Australia account. So in January each year, we'll look and go back and look at the performance information which has been collected on genotyped animals over the last 12 months, and rebates will be credited to their account if certain information has been collected. So in the case of, say, mature cow data, so mature cow weights, heights and body condition scores, if that had been collected on a genotyped female, then a $1 rebate per female would be paid back onto that member Australia's account. And then if that was done over 5 years on that same individual cow, then that would be up to a total maximum rebate of $5 to cover the total contribution. There's also rebates there for days to calving in a similar way, for some abattoir carcass data, and also for net feed intake. So the rebates aren't there for all traits, they're really for traits where the submission of that breeder data is of particular value to building that reference population, but they are designed to try and get a little bit more equitable situation where if members are investing in the collection of those hard to measure traits, then they're not being double dipping effectively. We're not asking them to contribute to that reference population twice. And there'll be further information distributed about that over coming months. The other part of the equitable access is around when base animals are genotyped. So base animals being animals without two known parents. In this case, they tend to often be animals where they haven't had female inventory paid, haven't been really indirectly investing in that reference population by contributing to Angus Australia. So in that case, there'll be a surcharge introduced of $25 when that animal is genotyped. So if a base animal is genotyped, in addition to the routine genotyping cost, then an additional $25 surcharge would apply to effectively cover the cost of accessing that genomic reference population. So they're just some of the different things which Angus Australia has been doing around, looking at how do we fund and make sure we have a genomic reference population available for use by Angus Australia members so that they can get the most value out of genomics and ultimately get the most accuracy, and therefore we maximise our rate of genetic progress. And also some things which we're looking around how equitable is that access and trying to put some things in place to ensure that everyone's contributing as they should. So thank you very much.